Hello everyone. In today's lecture we'll discuss about the various types of protein structures. Basically the proteins exist in four types of structures like primary, secondary, tertiary and quaternary. So now we'll see one by one. Types of basic structures. One is primary structure and second one is the secondary and tertiary structure and quaternary structure. So now we will see one by one what is the characteristic features of different types of protein structures So here is the primary structure of protein. So primary structure is nothing but it's a linear polymer formed by linking the alpha carboxyl group of one amino acid with the alpha amino group of another amino acid. So already we know that what is the linkage of this. So when alpha amino group binds with the alpha carboxyl group, it will form a peptide bond or amide bond. So in case of proteins we call it as peptide bond because it will be forming the peptide chain. So these primary structures are nothing but a unique sequence of amino acids that are linked by the peptide bonds to form a peptide chain. So these are the linkages between the alpha amino group of one amino acid and alpha carboxyl group of the another amino acid. So already we have learned that why we call it as alpha amino group, because it is attached to the alpha carbon. So this carboxyl group also attached to the alpha carbon or acidic carbon, that is why we call it as alpha carboxyl group. So when the bond is formed between the alpha amino of one amino acid and alpha carboxyl group of another amino acid, they will form a peptide bond. So they will form a linkage or chain kind of structure by joining all these kind of amino acids. So that linear chain which is formed with the help of peptide bonds, so that is called the primary structure of protein. So apart from these peptide bonds, there won't be any other kind of interactions in this primary structure. So that is why it is a very simple and primary structure, so which is having only peptide linkages which will be holding all the amino acids together in the form of linear chain. So the next level of protein structure is secondary structure. So secondary structure is nothing but a structure which is formed by the folding and twisting of the amino acids of the polypeptide chain. So here we have two types of secondary structures. One is alpha helix and second one is the beta pleated sheet. So in these both the structures, these structures or these proteins are held together in this kind of shape just because of the hydrogen bonds that are formed between the carboxyl group and amino group. So we know that to form a hydrogen bond we require a donor atom and an acceptor atom and a hydrogen atom. So here the hydrogen is present on the amino group, and amino group acts as a donor because it will be having the lone pair of electrons, so that is why it acts as a donor group, and carbonyl group, which is the electron deficient group, so that will act as a electron acceptor group. So with the help of these three factors it will form the hydrogen bonds between the amino acids of the neighboring amino acids. So here if you see the structure in secondary structure in alpha helix, so there will be a hydrogen bonding between the carbonyl of first amino acid with the NH2 group of the fifth amino acid. So because of this distance it will form, because of this interaction between the first and fifth amino acids, so it will form a twist kind of thing. Because of that it will form the helical structure. So in each turn of this helix, so there will be 3.6 amino acids that are present in each turn. So because of these hydrogen bond interactions it will form a ribbon kind of structures. So the second case is beta pleated sheet. In this secondary structure, another kind of secondary structure is beta pleated sheet. So in this beta pleated sheet what happens, so all the peptide chains are lined together parallelly. So then there will be a hydrogen bonding between the side chains. So one peptide chain amino acids and another peptide chain amino acid, there will be interaction, that is nothing but the hydrogen bonding between the carbonyl and amino groups. So because of this it will form a sheet kind of structure, and the R groups, which are the alkyl groups or any other groups, the R group subgroups will be projecting to the down and bottom of the sheet. So in these secondary structures, whether it could be alpha helix or beta pleated sheet, there won't be any interaction between the R groups. In case of alpha helix, so those R groups will be projecting outward the helix, and in case of beta pleated sheet they will be projecting down and bottom and top of the beta pleated sheet, but they will not take any part in the interactions. So these are the two types of alpha helix and beta pleated sheet. These are two types of secondary structures. So in beta pleated sheet there could be two types of beta sheets. One is parallel and second one is the anti-parallel. What is anti-parallel and parallel? So when the amino groups of all the peptide chains which are forming the sheets are coming one side, so then they are parallel beta pleated sheets. If they are alternate, suppose in the first chain alpha amino group is at the right side and in the second one it is left side, so it is that if you see the direction, if it is going towards the right side for first peptide chain and the second peptide chain is going towards the left side, then these are called the anti-parallel beta pleated sheets. So the thing is, remember that in the first primary structure there will be interaction only between the amino acids with the help of peptide bond. There won't be any other interactions, but in case of secondary structure you will be having the peptide bonds which is forming the peptide chain. Along with that there will be a hydrogen bond between the amino groups of the neighboring amino and carbonyl groups of the neighboring amino acids. So because of that we will be getting the alpha helix and beta pleated sheets. So next comes the tertiary structure. So in this tertiary structure it is nothing but the three-dimensional structure of the polypeptide chain. So we have already seen that in primary and secondary structure. In primary only peptide bonds are there, and in case of secondary we are having peptide bonds along with hydrogen bonds. But in case of tertiary structure, so here in what is the main difference is that here the R groups will be participating in the bonding interactions. So already we have seen when we were discussing about the different types of the 20 types of amino acids. There we have seen there are different types of side chains which will be having OH group or SH group or carboxylate groups. So those groups will be participating in the interactions or bonding interactions, or nothing, we call it as non-covalent interactions. So because of those non-covalent interactions, these peptide chains will be forming a three-dimensional structure. So there are various types of bondings which are present in the tertiary structures, like peptide bond already we have discussed, which will be forming between the amino acids and forming the peptide bond or peptide chain. And next is the disulfide bond. So this is also a covalent bond which will be forming between the sulfur atoms. So already we know that there are amino acids which will be having SH group, that is cysteine. So cysteine amino acids will be having the SH side chain. So when these cysteine amino acids are coming together, they will form a side SS double bond, that is nothing SS bond, that is not double bond, that is single bond, but SS bond that will form, and that bond is called disulfide bond. So next one is the ionic bond. Ionic bond will be found between the charged molecules. So here if you see the fourth category, that is NH3 plus and O minus. So amino group of one amino acid and a carboxylate group of, so these two are in the side chains, not the main amino group or main carboxylate group. When main amino group and main carboxylate group of the amino acids bind together, that is called peptide bond. But here this is a non-covalent interaction, so this is nothing but ionic bond. So this is not the covalent bond, and these kind of interactions are called ionic bonds. And next category is hydrogen bond. So already we have discussed, which is forming between the, if you see the first category here, so CH2OH is there, so between this OH and COOH. So here carbonyl O is acting as an acceptor, and this OH CH2OH oxygen is acting as a donor, because that will be having the lone pairs. But this carbonyl group is electron deficient, so because of that it is acting as a electron acceptor, and there is a hydrogen group in hydrogen atom in between, so that will be forming the hydrogen bond which can stabilize the structure. So here there are 5 types of things, and last category is hydrophobic interactions, which are present in the in between the alkyl groups of the different polypeptide chains, or between the R groups of different amino acids. So these are hydrophobic interactions. So combinedly all these 5 types of interactions will hold the amino acid groups of this peptide chain together, and they will be forming the three-dimensional structure, that is nothing but the tertiary structure. So here you can see the interactions and along with the three-dimensional shape of a tertiary structure of any polypeptide chain. So the next category is the quaternary structure. So here if you see the quaternary structure of amino proteins, so in previous cases we were discussing about the interactions between the amino acids of the single polypeptide chain in case of primary, secondary and tertiary. So there was only one polypeptide chain and there was an interaction between the amino acid groups of the same polypeptide chain. Thus, we were discussing about the single polypeptide chain. So but if the interactions are there between the two different polypeptide chains, so whatever structure they will be forming, that is called the quaternary structure. So the main difference is here we will be having different polypeptide chains and there will be an interaction between the different polypeptide chains. So whatever interactions that we have discussed till now, so those interactions will be there, but interactions will be present in between the different polypeptide chains. So the structure that will be forming, that is quaternary structure. So the best example for this quaternary structure is hemoglobin. So we all know the function of hemoglobin, that it will transfer the oxygen from lungs to body tissues. So there that oxygen will be utilized for respiration purpose, so that the entire process we will be discussing during the metabolism. So how it will, the oxygen will be utilized by the cells to get the energy. But for the time being, just you remember that these hemoglobins will be an example for the quaternary structure. If you see the hemoglobin structure here, it will be having the alpha chain and beta chain. So these are two different types of amino polypeptides. So there will be an interaction between these polypeptide chains, so that interactions will result in a quaternary structure of the protein. So this is the summary of all the four types of structures: primary, secondary, tertiary and quaternary. So just I will revise it once again, that in primary structure there will be the amino acids will be linked together with the help of peptide bonds and they will be forming a linear chain of structures. So that is nothing but the primary structure. And in case of secondary, we have two types of structures. One is alpha helix and second one is the beta pleated sheet. So alpha helix is nothing but a ribbon kind of structures which will be formed with the help of hydrogen bonds. And in case of beta sheets, that is also found with the help of hydrogen bonds, but here the polypeptide chains of same kind. So don't get confused with the quaternary structure. There polypeptide chains are different kinds, but in case of beta pleated sheet, same kind of polypeptide chains are arranged in a linear sheet manner parallelly. So there will be an interaction between the amino and carboxyl group in the form of hydrogen bonds. So next comes the tertiary structure. It is nothing but a three-dimensional structure where the R groups will be participating in the interactions. So there will be a non-covalent interaction between the R groups of the polypeptide chain. So if only polypeptide bonds are there, then we call it as primary, and if peptide bonds and hydrogen bonds are there, then we call it as secondary. And if along with these two, if there is any interaction between the R groups, then we call it as tertiary structure. And the last one is the quaternary structure. So here the interaction between different types of polypeptide chains. So the best example for this quaternary structure is the hemoglobin. So with this, your macromolecular analysis structural part is over. So in next lecture we will be discussing about the functions of proteins. So already I have discussed about the various functions of proteins during the biomolecules lecture, but here again I will revise once again and I will try to give some extra information about the functions of various types of proteins. So after this unit, then we will start the enzymes part.